As we're setting up the computer, I want importantly to start by thanking really everybody who organized and attended and presented at this remarkable conference. In particular, my mentor and friend, Asim Ansari, who is responsible for my not pursuing a career in English literature, but with whom I worked when I was an undergraduate at Harvard, and has had, as I think you'll see from this talk, a profound effect not just on my choice of a research field, but the way in which I'm approaching a very specific and maybe small problem in cancer biology. It has also been a maybe undeserved privilege to present this work, which is the second research project we've submitted for publication from my new independent laboratory at the Dana-Farber Cancer Institute. It is the work that I'll present is the labor of a first graduate student, Raymond Mollering, who's very much the hero of the success of this project. And then finally, I have to say, moving to Boston and living in Cambridge and working in Kendall Square, having grown up in suburban Chicago, it's never lost on me the unique opportunities to interact with personally real legends in the fields of science. And as a chemical biologist now, this is a great honor to show you our work, and I really welcome all of your feedback, Dr. Khurana. So the project that I'll present today is our efforts to target transcriptional regulatory complexes in a complex mammalian system. And I will perhaps today, quite prematurely, share with you what is the grounding theory of my small three-person, hardworking academic lab as regards cancer therapy of the future and our mechanistic studies of transcription in this disease background. So I provide for you first the Wikipedia definition of cancer, which I think everyone in the room is probably quite familiar with, and then the way in which we're interpreting cancer in our lab, which is a disease of cell state. That these malignant phenotypes that are so devastating to the patients that on Thursday mornings I still see are widely regarded as attributable to genetic alterations. And it's fair to say that 99% of all of the people in ligand discovery in cancer are interpreting the medical literature to tease out these genetic alterations and then fashioning small molecules that can approach these pathways. But we also note that these mutations, that these translocations always arise in the context of a particular and permissive cellular identity. The mutations that transform the committed lymphocyte progenitor to develop T-cell leukemia cannot support a myeloid leukemia. And so then, listening to the amazing work of the developmental biologists, some of which you just heard, and thinking about innovations in ligand discovery, new platforms, new chemistries, we try to bring convergent advances together to approach pathways of cellular identity, and in particular, those master regulatory complexes, transcription factors, so as to develop new tools for mechanistic study of gene expression, but ideally to seed new therapies for further development by those familiar with the process of drug discovery. So fundamentally, everyone is quite up to speed now about how gene expression works, that there are transactivating protein complexes that through mediators recruit coactivators and ultimately RNA polymerase II, and that this can be traced with discrete measurements of gene expression, but also now with genomic techniques. Building on the shoulders of Dr. Khurana's work, of course, has been said, that you can actually study whole pathways. And these are very helpful tools when trying to devise new small molecule inhibitors of gene regulatory complexes. Well, the lowest hanging fruit in gene regulation are certainly those regulatory proteins, typically enzymes, that modulate the function or the binding events, as Mark had told us, via enzymatic activities. And these, of course, have greasy pockets and enzymatic functions that are readily amenable to ligand discovery and small molecule optimization. And so it's in my training with Stuart Schreiber at the Broad Institute, I focused on these sort of lower hanging fruits so as to learn the discipline of ligand discovery. But targeting transcription factor complexes themselves with small molecules, which is the interest of our lab, is really a small and cottage industry because it's been widely regarded as a very difficult problem in industry and academia. And so the molecules that we do have access to in the clinic and in the laboratory are typically derivatives of human hormones or plant hormones that interact with hormone receptors in the nucleus, and then certain natural product toxins of RNA polymerase. It's important to point out that these molecules are very important cancer drugs. And so plausibly, if we could create new classes of molecules targeting these regulatory proteins, they too might also in the future be successful. And we're starting to see some glimpse of this, that there are molecules now that are targeting the chromatin-modifying enzymes, and those in boldface are compounds that I've had a chance to work on, targeting DNA methylation pathways, targeting transcription factor post-translational modification states such as JAK kinases, phosphorylating stats, et cetera. And I've done some work to discover new molecules that regulate the nuclear trafficking of these types of transactivators. But it's fair to say, on Thursday, when these patients with T-cell leukemia come into our clinic, we look at them in the face and we have a conversation with them that we couldn't have with my dad when he developed pancreatic cancer, and that is, we know exactly why you have cancer. 51% of the time, it's because of a mutation in the Notch complex and that pathway. And so as a patient or family member of that patient, you might say, well, give me the Notch inhibitor. It's frustrating to say, well, there isn't a Notch inhibitor, and why not? Well, I don't know, maybe it's hard, or maybe it's a low incidence cancer, et cetera. But everybody's studying Notch, and certainly for the scientists, we thought it would be quite valuable to start our studies in small molecule targeting of transcriptional regulatory proteins, focusing on this pathway. Although I want to point out that the richest targets in all of cancer biology, as best I can tell, are all transcription factors, because either they, with primary lesions of amplification or translocation, or as the mediators of upstream constitutive signaling pathways, they determine and maintain the neoplastic phenotype. And so these molecules are of pressing need, and we saw already from Dr. Dervin an exciting new class of molecules that, collaborating with Asim and others, have started to demonstrate real activity in cellular models of cancer. But what I'll talk about today is a higher hanging piece of fruit that we used a sort of lower hanging piece of chemistry to approach. And that are molecules that bind to and destabilize directly transactivation complexes, and that we might follow these molecules with their effects on transcriptional networks so as to gauge their activity, but also their pathway specificity. So I cut my teeth in Stuart's lab learning just some fundamental approaches, and in our laboratory now we like to think that we're trying to be as creative as we can with discovery platforms such as this effort to print small molecules in microarray format, not unlike oligonucleotides, so as to probe with standard microarray readers for novel interactions between transcription factors and small molecules. And all of this is published. You can read for yourself. You'd be the first person. But I think that this is a very powerful methodology to identify binding events to proteins of interest, unbiased for biochemistry, as you can actually use this platform with cracked cell lysates. Also spend a lot of time thinking about the biochemistry of the molecules or macromolecules that we're targeting, and we have significant collaborative work ongoing with Wei-Ping Tang here from the Department of Chemistry and Pharmacology to develop robust platforms of studying chromatin-modifying enzymes, and with these in hand, to develop focused libraries using biasing chemistry, sort of privileged scaffolds or pharmacophores, and then building out rapid diversity through efficient chemistries, one of which Wei-Ping developed while he was in Stuart's lab, to realize the most potent and selective, and now species selective, inhibitors of chromatin-modifying enzymes ever reported. We like to also listen to those in the computational biology literature and have a fruitful and ongoing collaboration with Olaf Wiest at my least favorite university, Notre Dame, and at Madison. I don't have to make that point emphatically, but that's just because of sports, not chemistry. We've been using computational approaches to try and understand the principles of molecular recognition, in this case of natural products, but as I'll show you in a moment, of these peptide inhibitors of the Notch complex. And apologize for the coloring of this, this figure from our first paper out of our lab, but we wrote it on the weekend of the Bears-Packers game. But I will say that I did a lot of medical training, and I think more and more, and my feedback, if it's at all appropriate to say it at this conference, is that the model of disease is now the human. And our ability to initiate human clinical investigation with real biological endpoints, be they genetic, chemical, molecular, computational, is quite a frame shift, I think, in basic science research. And we're always in our group meetings trying to figure out in what way we can bring our theories to the bedside. And with a project that's just recently out the door, we've identified a cancer that its molecular pathogenesis has been completely resolved by Christopher French at Brigham Pathology, a rare and incurable pediatric malignancy that fuses an unknown activating protein called NUT adjacent to a bromodomain protein called BRD4. And by hyperacetylating chromatin, working with Chris's lab, we've been able to recruit this oncogene, which is untargetable by any chemical means, but recruit it with just global hyperacetylation away from these poised foci, inducing such a potent differentiation effect that you can literally peel these cancer cells, and this is from a patient's lung tissue, right off the culture dish. And when a patient was airlifted from Syracuse, New York to Children's Hospital, Steve Sallin, who's our chief of staff, approached us to say, well, I read your grant, it was pretty good. Should we treat this patient with an HDAC inhibitor? And I thought, no, that sounds very reckless. He just had cardiac surgery and has low platelets and is anticoagulated. But he impressed upon me, as pediatric oncologists always do, the pressing need for therapies in these rare diseases. And so we wrote in 7 days a clinical protocol with full implicit support from Merck, the FDA, and our institutional review board, which I think is a record at our institution, which was approved. And after 4 months on therapy, this patient's significant tumor wrapping around and encasing the heart went PET negative. So we'd like to think that the research that we're doing will ultimately make it to the clinic, if not sooner. But I want to, in the remaining time, tell you about this Notch project. So I have this idea, as I said, of approaching cellular identity as the target. That even if the mutations still reside, if they lack the support, the ballast of the tissue in which they evolved to cause the malignant phenotypes of cancer, then they wouldn't maybe not be able to sustain cancer. I'm not sure this project actually proves this, but it's a good first step to getting there. So Notch, if you're not thinking about it all the time, is really a series of proteins involved in a pathway that were identified in 4 genetic screens in Drosophila that had the common phenotype of notched wings. What we now know is that Notch is a master regulator of cell specification. And it mediates a number of binary cell fate decisions. Do I want to be a T cell? Do I want to be a B cell? Do I want to be a glial cell? Do I want to be a neural progenitor stem cell? And I'll tell you a little bit about the 3-part components of the transactivation complex in a minute. Notch is initially synthesized in a pre-pro form and then undergoes significant post-translational processing, where eventually it presents itself to the cell surface and exhibits juxtacrine signaling. And this is just an amazing signal transduction platform because it's actually physical approximation of a neighboring cell that tickles Notch, rendering it biophysically more susceptible to cleavage by gamma secretase, at which point it flows into the nucleus and then induces a classic repressor to activator switching mechanism. It binds a repressor, and in so doing establishes an interface onto which the private coactivator mastermind will then bind. And this drives, in the case of T-cell leukemia, when constitutively activated, all the malignant phenotypes of this disease. But importantly, it's also observed in breast cancer, lung cancer, ovarian cancer, medulloblastoma. Well, John Astor at Brigham and Women's Hospital, where I did my residency, published a paper in Science in 2004 showing that mutations in this heterodimerization domain that facilitate cleavage and activation, irrespective of a neighboring cell, or mutations that truncate the degradation sequence in the PES domain that stabilize and cause this protein to accumulate, are pathogenic, are the cause of most patients with T-cell leukemia. And this really got my attention, but the project didn't really start. Well, I didn't have a lab actually at the time, so that was a part of the problem. But I was sort of minding my own business at the American Society of Hematology annual meeting when I saw Steve Blacklow's beautiful crystal structure presented, where cognate DNA is recognized by, in yellow, this repressor protein CSL. And then shown also here is the anchor and repeat domain of Notch itself, the intracellular component that traffics to the nucleus. And there's this groove. And this excited me, because the idea was that here in this transcription factor, there is a pocket into which we might, with this structural information, fashion a molecule. But this is a long span, and you know that's the problem with protein-protein interactions, how to target them. And so we wanted to use all the inferences we had access to from developmental biology principally, but also this crystal structure and the accompanying structure in the same issue of Cell and C. elegans, to try and tease out where the most valuable and likely spot to position a small molecule might be. Well, targeting alpha helices, as it turns out, has a field all unto itself. And they can actually be targeted with dominant negative spans of the peptides themselves. But if you excise a helix from a large protein, it loses its structure. And so chemistries have been created over the last 20 or 25 years to stabilize this secondary structure of extended peptides. And so we had a lot of choices of what type of chemistry we might go with. I'll tell you a little bit about the reasons we chose hydrocarbon side chain stabilization. So what we did is we looked for the regions of maximum buried surface area, and we thought, well, that might be a good place to start. And we looked to try and extend the helical register to capture maximum cationic potential, thinking that that might facilitate intracellular access. And then we zoomed in on this region, and we got a sense of the electrostatic interactions revealed by the crystal structure. And there were two residues on the surface of this idealized potential molecule that were solvent exposed and not incorporated or involved in any particular protein-protein interaction. And so there we thought might be the ideal place to position an alpha-alpha disubstituted non-natural amino acid incorporated with peptide synthesis that we do now on microwave with quite high yield. And then using Grubb's Nobel Prize-winning ruthenium catalysis, we can then close this cross-link, prepaying synthetically the entropic cost of forming secondary structure stabilizing the alpha helix. And the chemistry had thankfully already been worked out by Schaffmeister in the laboratory of Greg Verdine right up the street. So I cold called Greg Verdine, and I said, well, I'm this guy, this doctor, I have this idea, I have no lab. Would you be willing to make a couple of molecules? And Greg most generously said, I think this is a fine idea. I'll give you a graduate student to mentor with me, and this is Ray Mollering. So with Ray, all of our structural inferences aside, we mapped out a series of molecules that would tile the interaction domain of mastermind that might function then as dominant negative peptides. We synthesized these compounds and then used circular dichroism to assess their relative or predicted helicity. And what I find really gratifying about this is that the wild-type short-span peptide mastermind 1, which corresponds to the molecule I'll tell you quite a lot about, the stapled alpha helix derived from mastermind 1, exhibits low 23% helicity. When you incorporate the alpha-alpha disubstituted molecule, even before peptide stapling, this AIB effect confers increased helicity. But closing the cross bridge results in 94% helicity. The nice thing about making these molecules in a synthetic fashion is that we brought the tail of the peptide up so that we could append all kinds of functionality. PEG linkers for pharmacology, FITC to follow intracellular trafficking, biotin for pull downs, or settle caps to use in routine cell biology. And so we've shown now by immunoprecipitating Notch itself that we can pull down CSL, the repressor, as well as mastermind, and that we can compete off mastermind with this molecule SOM1. And this puts SOM1, to the best of our ability, we don't have a crystal structure yet, at, quote, the scene of the crime. We've then used fluorescence polarization to also confirm binding at this site and have established a 120 nanomolar binding interaction. We've confirmed this binding interaction as well by surface plasmon resonance. And these studies taught us something very exciting about the way in which this complex assembles, initially through a high affinity interaction with a RAM domain, and then closing with a low affinity ankyrin domain. I think Mark, if he had stuck around, might like this, because what it shows is that the bar for inhibiting transcription factors is low. 100 nanomolar binders can disrupt 1 micromolar interactions. In complex mixtures such as cellular lysates, we're able to pull down Notch with our peptide and able to pull down our peptide with Notch, buffered by all of the other protein components in cells. Does the molecule get into cells? Of course it does, or I probably wouldn't have been invited. We made a FTSE conjugate, and that seems to penetrate cells as well as enter the nucleus, and we can directly correlate this to the net cationic potential of the molecule, and we can quantify it using high-content imaging, establishing a dose and response behavior, as well as exhibiting temperature dependence, all consistent with an active transport. The confocal microscope at the Whitehead Institute showed us that indeed this is entering through a vesicular pattern of endosomes and entering the nucleus. But would it do anything to gene expression? We used reporter gene assays initially, and what we found is that this idealized SOM1 peptide is quite active, the upstream peptide SOM2 a little less active, and everything else completely inactive, which validates this discrete binding interface of interest to us. And all of these experiments were performed with an inactive mutant. Well, in just the last couple slides, I'll tell you what I find most exciting, and that is that irrespective of any engineered system, in cultured human T-cell leukemia cells, this molecule downregulates target genes by both RT-PCR as well as the entire Notch transcriptional program by gene set, as determined by gene set enrichment analysis. And not only can we find the Notch signature, but embedded within the Notch signature, we can find those signatures of transcription factor networks linked to Notch, such as MYC in this case, but not Wnt. It kills T-cell leukemia cells that are driven by Notch, but has no effect on leukemia cells such as these chronic myeloid leukemia cells that are driven by BCR-ABL. It induces apoptosis. In order to publish this in a really splashy journal, we thought we had to go in vivo. Well, we didn't think so, but a reviewer said we had to go in vivo. And this was a challenge because we're a chemistry lab. And so we teamed up with Gary Gilliland, who's a brilliant leukemia geneticist, to build a bioluminescent model of T-cell leukemia. And I won't go through the details, but suffice it to say that by using a mouse with constitutively expressed luciferase in every tissue, we performed what I do on Thursdays all the time, a bone marrow transplant. We took the bone marrow cells out of this animal, transformed them with the human allele that causes leukemia, and put them into a syngeneic animal that's not luminescent. And what this led to was a significant burden of disease, and over about 40 days, they glow in the dark, full of leukemia. They respond to dexamethasone just like patients, and then they fail to respond to dexamethasone just like patients. But unfortunately, the molecule was not active when administered once daily. But we're not a drug company, so we scaled it up and we gave it twice daily, and all animals respond. In this model, we've been able to show also with gene set enrichment analysis in a pharmacodynamic-like way that in vivo, we're also targeting this Notch pathway. And we've made a version of this for flies that notches wings. So in conclusion, I present this first example in our lab of a small molecule that binds to and destabilizes a transcriptional activation complex that prevents a repressor to activator switch, and that further reduces this large interface of a key target in cancer and developmental biology to a much more tractable interface that one might approach with small molecule chemistry. And if you let me, I'll just make one last sort of plea to loop more of you into cancer research, and that is that this is still a real problem. That the overall prognosis of cancer has not changed substantially over 30 years. The number of new drugs is falling, and Mark Potashny's right. The genetics, if they've taught us anything, show that it's impossibly complex, and it will be very hard to approach with targeted therapy to benefit most of people with solid tumors. This is Ray. He's looking for a postdoc. This is my email if you want to get in touch with him or any of us. And again, thank you for the invitation.